delight to have Professor Laurie Isom from the University of Michigan with us today. Laurie has actually a deep emotional connection to Washington University because she did her undergraduate study here in the departments of chemistry and biology both. And then went south to Vanderbilt to do her Ph.D. And from there north again to even further north to Seattle, where she did a postdoc in Bill Catterall's laboratory, and really did seminal and beautiful work on the sodium channel and its beta subunits, including cloning and sequencing. Beautiful papers. And then the University of Michigan. She wears another hat, which is assistant to the Dean of the Medical School for graduate education, graduate studies. So that's on the educational format. And I think, to me, the important thing about Laurie's work is that she really looks at the complexity of systems at different scales. One of them is, of course, the ion channel and interactions between alpha and beta subunits, and the whole complex of the channel. There is not just one subunit or one protein. But this also carries over to the larger system, which is brain and heart, and in particular we talked today about epilepsy and sudden death from arrhythmias and how these could be connected. So that connection, I think, is something very important. It's recently been getting more and more recognition. So thank you for coming.
Thank you so much. So I'd really like to thank you all, especially Yoram, for the very kind invitation today. I'm honored to be back here at my alma mater. I cannot believe the building that has taken place on this campus. I didn't think you could go any further, but you did, and you're still going, and it's wonderful. So I have had a great time talking to old friends, making new friends, and setting up some potential collaborations. So I look forward to additional work with you. Now today I'd like to tell you a story, and before I do that, I'd like to relate to you an experience that my friend had with her child. It'll set the stage for the disease that I'd like to talk about today, called Dravet syndrome. My friend, her name was Lori as well, had a beautiful little girl, normal birth, uneventful. At about the end of the first year into the second year, she was pushing her in a shopping cart when she had a grand mal seizure and collapsed in the cart. So a number of seizures later and a number of emergency room visits later, she was diagnosed with Dravet syndrome. Dravet syndrome is really a perfect storm. It's a catastrophic pediatric epileptic encephalopathy that is largely caused by mutations in a sodium channel gene called SCN1A. And I'll tell you a lot more about that today. So children are born normally. Toward the end of the first year of life, they start to seize. There are multiple etiologies of the seizures. They all have developmental delay, cognitive decline, mental retardation, problems with gait, ataxia, other symptoms, sleep problems, autism spectrum disorders. It's really a huge cataclysmic event. So individuals with Dravet syndrome have a higher incidence than normal of undergoing SUDEP, sudden unexplained death in epilepsy, that I'll talk about more today, that we think has to do not only with disorders in the brain that cause seizures, but also disorders of the autonomic nervous system and disorders of the heart. And that's because the mutant genes that are involved in Dravet syndrome are expressed in multiple excitable tissues. So I'll be talking about that story today. This is an intractable epilepsy. So drugs that we all think about that really mainly target sodium channels that treat epilepsy are either don't work at all in Dravet syndrome, and in some cases actually make the seizures worse for a reason that we don't really understand. And Lori, my friend, and most of the mothers that I know with children with Dravet syndrome have their children on what they call the diet. Okay, and that's the ketogenic diet. And it's a very, very carefully controlled combination of lipids and proteins and almost no carbohydrate that for a reason that none of us really understand is the only thing that controls the seizures, but doesn't always. So the other day, my friend Lori told me that they were on their way to a special event at Disney World to raise money for Dravet syndrome, and on the way her daughter had a series of grand mal seizures in the car. And she said, luckily we got on the plane. These are the most resilient mothers ever. So what's happened in my laboratory that started out as a basic story, as Dr. Rudy told you about cloning and sequencing and understanding the structure and function of sodium channels, has now become a translational project in which I tell my kids in my lab, we have patients waiting. So we're trying to understand the mechanism, and in doing so, come up with potentially novel therapeutics that are different than what we normally think of to treat epilepsy. So as I told you, Dravet syndrome is caused by mutations in genes that encode voltage-gated sodium channels. And actually Dravet syndrome, oh wait a second, I'm supposed to do this. Sorry, I failed. Okay. Dravet syndrome is part of a spectrum of epilepsies that are the genetic febrile epilepsies and associated mutations. So many of these epilepsies begin with a febrile seizure. Not always. Lori's daughter did not. So but many times they do. Dravet syndrome is here, and you can see that there's some overlapping syndromes. And Dravet syndrome is caused largely by genes that encode voltage-gated sodium channels and sodium channel beta subunits. But today, in the first part of my talk, I'm going to concentrate on mutations in the gene SCN1A. So SCN1A is shown here. SCN1A encodes the voltage-gated sodium channel NAV1.1. And that's expressed all over in the brain and in the autonomic nervous system, in the peripheral nerves, and in the heart. So the majority of cases, 70 to 80% of Dravet syndrome cases are caused by mutations in SCN1A that cause haploinsufficiency. And you all may be looking at this map of a collection of mutations. This is from 2010. Now there are over 700 mutations that have been identified in SCN1A. How can all these mutations have the same result? Well, it's because they all cause loss of function. So haploinsufficiency, meaning the loss of one allele of SCN1A, is enough to cause this devastating illness. So how do we study this? Well, as Dr. Rudy said, early on we were cloning genes that encoded ion channels. We expressed them in a heterologous system, and we looked at their function. Now here's a representation of a mutation in the ion channel that causes changes on a patient's EEG. And we studied, and actually learned a lot by looking at heterologous systems like Xenophus oocytes or transfected mammalian cells. But we learned fast that we could not recapitulate the situation in a native neuron or native cardiac myocyte using heterologous systems. So many of us turned to transgenic mouse models. And again, we learned a lot. For example, my previous postdoctoral mentor, Bill Catterall, actually made the first mouse model of Dravet syndrome with a knockout, a null animal of SCN1A, and he looked at the heterozygotes and noticed, first of all, he found out that they seized. This is before we understood about the haploinsufficiency, so he was the first one to observe that. And what he reported now back in 2006, that when they acutely isolated hippocampal neurons, and here they isolated bipolar neurons and pyramidal neurons, and they just did this by morphology, by looking at them, and they measured whole cell sodium current. They saw no difference in the current in the excitatory pyramidal neurons, but they saw a loss, a major loss, in the sodium current in the bipolar neurons. And with, interestingly, no difference in the voltage dependence of activation or inactivation, there's the simple loss of functionality. And so he suggested that Dravet syndrome could be caused by disinhibition, right? So loss of sodium current from inhibitory neurons causes excitation, okay? Right. So we wanted to go a step further because mice are not people. What really happens in children? And so we decided to use this novel technique, induced pluripotent stem cells, in which we take skin biopsies from children with Dravet syndrome or normal non-epileptic control patients, and then reprogram them to make neuroprogenitor cells and then patient-specific neurons. And by we, I mean our translational team that involves a number of very talented postdocs and students in 3 laboratories, not only my own, but Jack Parent's laboratory. Jack is a neurologist and a stem cell biologist at the University of Michigan, and Miriam Meisler, who is a human geneticist at the University of Michigan. So the first patient that we biopsied was a 7-year-old patient with Dravet syndrome, and I'll refer to this patient's cells as DS1. This patient had a splice site mutation that took out exon 14, okay? And so you can see that on the RT-PCR here, this patient had 1 wild-type allele and 1 mutated allele. And here's a control patient with a wild-type allele, and the exon 14 takes out this section in black here of domain 2 of NAD 1.1. And then what it does is it makes a non-functional channel. We tried that in my laboratory. So if we made the mRNA and injected it into the Xenopus oocytes, we got no sodium current at all, as opposed to the wild-type allele that gave full sodium current. And then when we expressed it in HEK cells, we saw little to no protein when the mutant channel was expressed as opposed to the wild-type channel. So just like was predicted, this causes haploinsufficiency. And it's thought, and there's quite a bit of data now to show that this happens through nonsense-mediated decay of the mutant mRNA. Okay, then we collected a biopsy from a second patient, which I'll call DS2. This was also a 7-year-old patient with Dravet syndrome and uncontrolled epilepsy. And this actually was a nonsense mutation here that inserted a stop codon, a termination codon, and this also makes a non-functional channel. So 2 different mutations with the same disease. And in Jack's lab, he reprogrammed the patient fibroblasts into induced pluripotent stem cells, and he did this with this cocktail of transcription factors, c-MYC, PLF4, OCT4, and SOX2. Now, since then, this has fallen out of style, and we now do episomal reprogramming, so we don't have the retroviruses. But at this time, this is how our reprogramming was done. Yulu, who is the postdoc in Jack's lab who does this reprogramming, then proceeded to along the pathway of neural differentiation of these cells, and he saw differential neural rosettes expressed, and some markers, including PAC6 and Musashi, of the neuroprogenitor cells. Then he went on to neurodifferentiation, and he found, and with this protocol, although we have now modified it since, he got approximately 90% GABAergic neurons and 10% glutamatergic, and he learned that by staining for GABA and B-glut. We got a very small percentage of TH positive neurons, but the huge majority were GABAergic with some glutamatergics. The iPSCs that we made showed a forebrain regionalization, which is important because patients with Dravet syndrome have cortical thalamic seizures. So it's important that we have forebrain regionalization and not hindbrain. And here in this RT-PCR array are a number of forebrain versus hindbrain markers, and we convinced ourselves that we had forebrain differentiation and not hindbrain. For example, in Braille here is a hindbrain marker, and it was not evident in our iPSCs, while PAC6 and forced DS1 were. Importantly, they express sodium channels. So this is an RT-PCR for a number of sodium channels. You can see the gene name and the protein that is encoded here, and they were positive. And importantly, because Bill Catterall's data suggested disinhibition by loss of sodium current in inhibitory neurons, we had the classes of inhibitory neurons, and we stained for that, and we were able to convince ourselves that those inhibitory neurons were present. Okay, so off they traveled across the way to my laboratory, and Luis Lopez Santiago did a single-cell electrophysiology on these neurons. And you can see here some examples here of a pyramidal neuron and a bipolar neuron that he recorded from. And he looked at whole cell sodium current, and he did lots of experiments to convince himself that he had proper voltage control, that everything was controlled in these experiments. And he saw an increase in sodium current density. You can see that here for DS1, and DS2 was even larger. And strangely enough, you saw this increase in both the excitatory and the inhibitory neurons. So you can imagine our reaction. I said, Luis, this can't be right because Bill showed a loss of current. Well, we really convinced ourselves that we had an increase in current. And one of the ways we convinced ourselves was then Yukun Yuan, who's a slice physiologist, and he looks at synaptic and action potential firing, took them next. And he learned that the DS patient iPSC bipolar and pyramidal neurons had significantly reduced thresholds for action potential firing. And they fired repetitively with increased frequency. So you can see a control patient neuron here and a patient and a DS patient here. And I include the tables to show you that all the parameters were increased in the bipolars and the pyramidal neurons. And here's the important take-home message, that they spontaneously fired. And so, and this I'm showing you is I think this is a bipolar neuron, but this happened for all classes of neurons in the dish. So we basically have epilepsy in a dish. We have patient in a dish. We have these children's neurons that have been differentiated. They spontaneously fire. They exhibit epileptic hyperexcitability. And now we can use these as our substrates for drug development and to understand the mechanism of this disease.
Can I ask you something?
Sure. Yeah.
Can you go back to the slide?
Sure.
So how do you know what kind of cell this is? You say bipolar.
Yeah.
And then what?
So in our initial experiments, we could only use our eyes to look at the morphology. What we have done since then is after the recording, we suck out the contents and do single-cell RT-PCR to verify what these are. And we've also developed a series of fluorescent markers. So we have a synapsin M-cherry, so that M-cherry fires off the synapsin 1 promoter to tell us that these are actually neurons, and then for the subclasses. Because one of the problems is astrocytes can hide out as neurons, and they also have sodium current. And you find out a lot of these bipolar neurons are actually astrocytes, but now we've convinced ourselves by these additional experiments that they're actually the neurons that work.
So do you think they're capillary? That's what you said?
Yes. Yes. Okay.
And do you have to do anything to enrich in those forebrain type of cells? Because it's not surprising that, for example, when one derives the cardiac myocytes from iPS cells, you have every type of cell.
We do. So we have worked on this, Jack has worked on this protocol to really perfect it. Now he gets about 50-50 of the bipolars and the pyramidals, and he knows their cortical, okay, from a number of studies. We still get, in the background, some undifferentiated progenitors. And the way we get rid of those now is by labeling with the synapsin M-cherry. So everyone that we want is red, okay? And that's true for the cardiac myocytes too. You just have some undifferentiated stuff in there. Okay, and the table to show you that both pyramidal and bipolar neurons are affected by this DS mutation. And remember, both patients and multiple lines from both patients were equally affected.
Quick question, how do you, because we're always wondering about that, how are you determining the resting potential in the spontaneously active neurons?
So we determine the resting potential upon break-in before we get much dialysis, okay? So I can show you the protocol.
But it's more depolarized, right?
The DS neurons are slightly more depolarized than the controls, and that was consistent across. And actually, the pyramidals were slightly more depolarized than the bipolars. Since this time, excuse me, since our paper was published, another paper from a group, from Dr. Gao's group in China, was published with Dravet syndrome iPSC neurons, where they preferentially made pyramidal neurons, excuse me, and they saw a similar hyperexcitability and increased current. So why? We have haploinsufficiency, we lose one of our sodium channel alleles, and we get more sodium current. How does that happen? Okay, so we're working on this. And so far, here's what we found. When these two postdocs in my lab, Will Brackenberry and Heather O'Malley, have stained the immunofluorescence for sodium channels in these differentiated neurons, we first looked at NAD1.1, that's the channel that's mutated, and NAD1.2, and we didn't see a difference. Now, sodium channel antibodies are not good enough to see a twofold decrease. So you should see half of 1.1 that each with the antibodies in their current state. So here's what we see for NAD1.1 and NAD1.2. But what we did see that was very interesting, that the control neurons at this stage of differentiation have little to no NAD1.6. NAD1.6 is expressed in more mature neurons in the brain, and it appears that it is not expressed in the control patients, but it is expressed in the Dravet syndrome patients. Now, to follow this up, we made an shRNA virus that specifically knocked down SCN8A. Here's the vector. And we tested it first in transfected HEK cells. So in HEK cells that express NAD1.1, we saw a significant knockdown of the current. In HEK cells that express NAD1.6, a different, I'm sorry, that is backwards. Okay, I have to change this. NAD cells that express NAD1.6 encoded by SCN8A were specifically knocked down, whereas cells that express NAD1.1 were not. I'll get to that. And in the iPSC neurons, we got a significant difference in the current. So here's the important experiment in iPSC neurons that were either treated with a scrambled virus or the SCN8A shRNA. The hyperexcitability returned to control levels. So we were able to bring the sodium current back down to control levels that were comparable to controls. And we also returned the excitability to something that appeared to be normal. And a growing body of evidence suggests that SCN8A gain-of-function mutations result in epilepsy and SUDEP. And the most recent was a paper from our collaborator Michael Hammer, who showed that a gain-of-function mutation and SCN8A in a child with a Dravet-like syndrome was responsible. So there's a growing body of evidence that this increase in SCN8A expression could underlie the hyperexcitability that is seen in Dravet syndrome. So of course this is not the end of the story, and we have a lot more work to do to prove that this is the mechanism, and if so, how does haploinsufficiency of one channel translate to overexpression of another channel?
Excuse me.
Yes.
So in those records, there's a huge change in the ankle resistance. So does that mean that there's a window current of the sodium current, and that's why is that what
We
you
haven't seen a window current, but I understand what you're talking about. There is a difference.
huge, actually.
yeah. And I don't know the answer, Jane, but that's, yeah, thank you. So here's an important question. If you are a neuron that grows up on glass, as these iPSCs were, they were differentiated in a dish, not in the brain, are you modeling the situation that happens in the hippocampus, in the cortex, in situ, in the developing brain? This is a very, very important question for the entire field, not just our particular project. So we're working on this, and I don't know the answer, but I want to show you where we are. What we do is we differentiate iPSC neurons to the neuroprogenitor stage. We label them with a fluorescent marker, and we cannulate a rodent's brain, and we implant the neuroprogenitor cells inside in the rodent brain. We sew up the rodent brain, and we allow the pup to develop. We're still deciding how long the pup should develop, but we're working on it. And then we isolate the brain and we make slices, and we actually find the iPSCs that are glowing, okay, in different parts of the brain. You can see one here, CA1, and in other parts of the brain. And then we go ahead and record from the cells that are labeled. And this is just to show you that we can actually record a family of currents. You can see this inward current here, followed by an outward current. They're kind of immature at this stage. But we're working on this important experiment because these cells that have been implanted in the brain should also show hyperexcitability if our model is correct. So hopefully, next time I talk to you, I'll be able to tell you what we found. So to summarize up to this point, Dravet syndrome is a severe epilepsy largely caused by SCN1A sodium channel mutations resulting in haploinsufficiency. We derive 4 brain-like neurons from 2 Dravet syndrome patients and 3 control patients. Pyramidal and bipolar Dravet syndrome neurons from both patients, different genotypes with the same disease, showed increased sodium currents and epileptic-like hyperexcitability. This has now been repeated by another laboratory using the iPSC technique. And so far, upregulation of SCN8A encoding NAD1.6 suggests a novel epilepsy mechanism. So let's get back to Dravet syndrome. Kids with Dravet syndrome have a very high incidence of SUDEP, sudden unexplained death and epilepsy. And we're very concerned with what the mechanism of this could be. So sudden unexplained death and epilepsy is defined as non-traumatic, non-drowning death in patients with epilepsy, excluding cases of status epilepticus. And SUDEP accounts for a large percentage of deaths in epilepsy, and up to 50% of kids with Dravet syndrome die spontaneously from SUDEP in early childhood or adolescence. And indirect evidence has linked SUDEP to respiratory problems, dysregulation of cerebral circulation, cardiac arrhythmias, disruption of respiration. Actually, it's probably all of those things in one. So we were struck by the fact that SCN1A, encoding NAD1.1, is expressed in heart in addition to brain.
So Laura,
Yeah.
is the sudden death associated with an epileptic episode or not?
So indirectly, so the epilepsy has to be a factor. Sometimes the death closely follows a seizure. Many times it does not.
So it could be isolated,
It
no
could be
epileptic
isolated.
episode,
Yes,
just sudden death.
yes. Many
Does it
times.
occur under certain conditions like exertion, or does it have emotional stress, or does it happen at rest?
Most of the time it happens during sleep. Very similar to long QT3 or Brugada. There have been a few sleep studies with epileptic patients now to try and catch near SUDEP events, and a few have been reported. They will all have been at night during sleep. Yes?
Are these incidents also always connected to like the genetic mutations in certain channels, or?
No. We use Dravet syndrome as a model because it's close to a monogenic disease that we can actually follow. But all epilepsy patients, whether it's genetic epilepsy or acquired epilepsy, have a very high risk factor for SUDEP. So it's something that happens secondary to a seizure. And so we're starting by looking at the genetic epilepsies because it may be easier to look for a mechanism. But we hope that then we can extend that to more generalized epilepsies. So we were struck by the fact that SCN1A encoding NAD1.1 is expressed in both heart and brain. And this was shown by Bill Catterall's laboratory and our laboratory. NAD1.5, that is the predominant sodium channel in heart, that you all think of as the cardiac sodium channel, is largely expressed at intercalated discs, and there's some expression on the lateral membrane. TTX-sensitive sodium channels, including NAD1.1, NAD1.3, and NAD1.6, are known to be expressed at the T-tubules. And in fact, my collaborator, Mario Delmar, with his super resolution scanning microscope, can now record sodium currents right at the triad of the T-tubule, and that is all TTX-sensitive current. So we asked, is there anything going on in the heart in a model of Dravet syndrome with SUDEP? And we proposed that a mechanism of SUDEP and Dravet syndrome is mutant channels expressed in both brain and in heart. So Jack's lab has been making IPS cardiac myocytes for us. And here's just some examples of the spontaneously beating cardiac myocytes from 3 of our patients. And David Auerbach in my laboratory has been recording sodium currents from these dissociated cardiac myocytes. Now the same problems are evident here with maturity and heterogeneity. But, of course, early days, we were excited to see versus control non-epileptic patients an increase in sodium current in one of our clones of the IPS cardiac myocytes. However, another patient, the DS2, and this is the one, they both had increased current in the neurons. Only one of them had increased current in the cardiac myocytes. Now, it could be that it's a technical issue, or it could be that maybe this is a biomarker, maybe it could be predictive for patients with Dravet syndrome that may be at higher risk for SUDEP than others. We don't know. So we continue to work on this and to improve our ability to make more mature cardiac myocytes. But in the meantime, we asked, do mice that are DS model have a cardiac phenotype? And we used not Bill Catterall's model, but Yamakawa's model, where he knocked in a human mutation, this one here, R1407X, which knocks a stop codon in right here in domain 3, and it kills the channel and it makes haploinsufficiency. So these mice have one functional allele of SCN1A and one mutant allele, and they undergo SUDEP. So here's the survival curve, and about 20 to 30% of the mice undergo SUDEP, and the majority of them undergo this death in the first, let's say, 2 to 3 weeks. And we measured a group of mice with implanted telemeters, and we saw that of the DS mice, 3 of the 13 died spontaneously during our experiment. And importantly, these mice were allowed to come to equilibrium for 2 weeks and to regain their circadian rhythms following the implantation of the telemeters. So David Auerbach again measured their sodium currents in isolated cardiac myocytes, and he found an increase in sodium current. And he found this in the transient current and in the persistent current. And he saw a slight but significant hyperpolarizing shift in the voltage dependence of availability or inactivation, and voltage dependence of activation, consistent with an increase in NAD1.5 currents that are more negative than the TTX sensitive current. He also saw no difference in current when he applied 100 nanomolar tetrodotoxin, suggesting that there were no differences in TTX sensitive current, but differences in the TTX resistant current. The DS myocytes exhibited increased excitability and incidence of early after depolarization. So he saw a decreased stimulus threshold for action potential generation. You can see that here in the DS mice. He also saw increases in the AP upstroke velocity, and importantly, he saw an increased incidence of early after depolarization. So you can see that in 2 of 11 cells measured here for the wild type, and 11 out of 17 cells measured for the DS myocytes. And by the way, these are ventricular myocytes.
This
yes.
earlier after depolarization, they start from pretty low. Not sure what's the scale, but they look like they start from pretty low voltage.
They start, okay, so yes.
It's not really sort of, high level. It might be related to calcium cycling.
It might be related to calcium cycling. And in fact, calcium handling is something that we'd really like to look at. In these science we're getting ready to look at that. So when we looked at the ECG parameters of these mice, we saw that they had a prolonged QT interval. And you can see this here for the QT 50, 75, and 90, and then the corrected values that are all significantly higher in the Dravet syndrome model mice than the wild type littermates.
What age?
These are P21. We implanted them between P21 and P24, and we let them go for up to 60 days on telemetry. For the 3 that died, we paid particular attention to their heart rate that preceded death. And you can see that here for 2 of the mice. There were 2 of the mice that died spontaneously, DS1 and DS2, and DS3 became so moribund that the animal facility ended up euthanizing the mouse. So you can see that there is a bradycardia that happens during the last hour or so prior to death, that then shoots up into a tachycardia and then ends up in the catastrophic event. And that happened as well approximately 24 hours prior to euthanizing the DS mouse. We also saw increases in heart rate variability just prior to death in the DS1 and DS2 mice that died spontaneously, and then the DS3 mouse that became moribund and we had to euthanize. You saw this great difference in heart rate. We have also been analyzing heart rate variability and trying to look at autonomic innervation in these mice. So far, we don't see differences in overall autonomic tone, but we do see differences just prior to death in those intervals, the last hour, the last few hours prior to death, where we see differences in the balance, the sympatho-vagal balance. We don't have those data ready yet to share with you. So, up until here, I showed you that DS patients are at high risk for SUDEP. And DS mouse cardiac myocytes have increased the tetrodotoxin-resistant sodium current compared to their wild-type littermates, and they have evidence of arrhythmia. Preliminary data suggests that the patient-specific DS myocytes also have increased sodium current, but that's more complicated, and it's different than the genetically identical mice that are the DS models. And we asked whether SUDEP may be an arrhythmia that occurs both in brain and in heart.
So did the arrhythmias, I mean, if you had the recordings, right, from the
Yeah.
telemetry, was it you said it was a tachyarrhythmia right before they died, but was
Right.
that like a long QT, torsade type of arrhythmia
Right.
that degenerated into VF, or did they really Brady down and then just have this end kind of a
Yeah.
catecholaminergic surge?
So both things happened. So right before death, we saw this lethal bradycardia. Before that, we saw incidences of ventricular fibrillation, which when we measured we measured by fast Fourier transform, we saw a characteristic 25 hertz frequency there. So we called it VF. And it didn't have an associated seizure that we were able to observe, although we did not have implanted EEG electrodes, so we could only tell you that by eye. Okay? So they would go in and out of VF, and then at the end, they ended up with this bradycardia. So there are more than one gene that can be responsible for Dravet syndrome. And I talked to you about the SCN1As, but SCN1B is also, although more minor, can be mutated and be responsible for Dravet. So can we glean something from what we know from these patients and these models? So you probably know, and this is my favorite part, is that voltage-gated sodium channels are heterotrimers. And not only are they composed of this large alpha subunit here that you can see on the western at 260 kilodaltons, but they are associated covalently with the beta 2, or now we know beta 4 subunit by disulfide bonds, and you can see that here, or non-covalently by a beta 1 subunit, you can see here. And recently, we learned that the gene encoding beta 1, SCN1B, has another splice variant called beta 1B that is secreted. So it doesn't have a transmembrane segment, and it's found in the extracellular media. Now, mutations in SCN1B encoding beta 1 can cause Dravet syndrome too. In my laboratory, we spent the last 20 years figuring out what beta 1 subunits do. And we learned that they signal through multiple pathways on multiple timescales. For example, if we express alpha subunits and zenoprazoocytes alone, we get a sodium current that activates and inactivates slowly. But when we co-express beta 1, you can see that the rate of inactivation becomes much faster. The voltage dependence of inactivation shifts in the hyperpolarizing direction, and the sodium current density is increased. And we learned that that's through chaperoning of alpha subunits to the plasma membrane by beta 1 subunits. We also learned that beta 1 subunits are cell adhesion molecules of the immunoglobulin superfamily, and they can undergo homophilic and heterophilic adhesion. You can see an example of homophilic cell adhesion here from beta subunits on 2 adjacent cells that result in cell-cell junctions or cellular aggregation, and the result of that is the recruitment of the anchorin cytoskeleton to points of cell-cell contact. Beta subunits are also cleaved sequentially by beta and gamma secretase, similar to amyloid precursor protein. And we know that this intracellular domain of the beta subunit can translocate to the nucleus and affect sodium channel alpha subunit transcription. So you can see here how beta subunits are fibroblasts that don't express beta subunits, do not aggregate, whereas cells that express beta subunits aggregate. And, importantly, neurons that are grown on a fibroblast monolayer that don't express beta 1 subunits have a certain amount of neurite outgrowth, but that amount of neurite outgrowth is multiplied by 2x neurite outgrowth, when beta 1 is expressed in the substrate. And we show that this is through homophilic cell-cell adhesion. We have a beta 1 null animal, and I think you can't see that very well, but one of the things that beta 1 does is chaperone sodium channels to the correct subcellular domain. So you can see in the control here that NAD1.6, I'm not sure you can see that, is expressed at axon initial segments, with anchor G as the marker, but in the beta 1 null, a significant proportion of axon initial segments do not express NAV1.6. So beta subunits chaperone alpha subunits to the right place. And finally, in collaboration with Jane Bun, we learned that beta 1 associates with potassium channels. So beta 1 is a busy molecule, and it governs, so to speak, a multi-subunit, multi-molecular complex of sodium and probably potassium channels to bring important molecules to this complex. And without beta 1, you have changes in sodium channel expression, sodium current, sodium channel localization, and neuronal pathfinding in the brain. So we know that beta subunit mutations result in epilepsy in humans. And the first mutation was found in this immunoglobulin loop domain that's responsible for cell-cell adhesion, this C to W mutation. And that's an important mutation because that's the disulfide bond that holds this IG loop together. And we showed that cell-cell adhesion is inhibited by this mutation. And that led us to suggest that these cell adhesive functions of SCN1B or beta 1 could be clinically relevant, especially in epilepsy. So we were the first, we meaning my MD Ph.D. student, Gustavo Coutinho, showed that a loss of function mutation in SCN1B, this R to C mutation here, when homozygous, resulted in Dravet syndrome. So this patient that we worked with was a homozygote for this mutation from consanguineous parents. And this patient died of SUDEP and uncontrollable epilepsy at the age of 13 months. Recently, another patient was described by our collaborator Yamakawa, and this patient was also homozygous for this mutation, I to F, again in the IG loop. This patient inherited 2 copies of this mutation, and the patient has a very similar disease. We also learned that the splice variant of beta 1, called beta 1B, that is secreted, causes epilepsy in 3 patient families, and does so by this mutation causes beta 1B to be retained inside the cell. So both splice variants of SCN1B are linked to epilepsy, including this very catastrophic epilepsy, Dravet syndrome, that ends up in SUDEP. So we learned that our SCN1B null mice are models of Dravet syndrome, and actually this list of phenotypic characteristics of these mice is very, very similar to children with Dravet syndrome. And here's a model of 2 of our mice. You can see one here that is a null, an SCN1B null, and here this is also an SCN1B null having an absence seizure. So these mice are born normally. They begin to seize around postnatal day 10, and they die of SUDEP by around postnatal day 21. We've never had a mouse live longer than postnatal day 26. So I'm going to tell you a little bit about them, but they have everything wrong, including disrupted sodium channel expression, disrupted currents, disrupted potassium currents, hyperexcitability, neuronal pathfinding. It's a catastrophic disease. We measured an SCN1B null brain slices are epileptic. So you can see those here. Some wild type EPSCs in cortical layers 2 and 3. You can see the wild type and the null. Okay? And population spikes in CA1 in response to Schaefer collateral stimulation, in the wild type and the null. And different areas of the brain are more epileptic than others. The neocortex is the most epileptic area of SCN1B null brain. The SCN1B null neurons have a lower threshold and higher firing frequency compared to the wild type. I think you can see that here in a series of recordings in the wild type brain versus SCN1B null. In addition, SCN1B null neurons are spontaneously epileptic. so you don't have to stimulate them, just record, and especially in the cortex, you can see the spontaneous hyperexcitability. They look a lot like our iPSC neurons. Well, sodium channel beta 1 subunits are expressed in brain and in heart, and they're associated with both the TTX sensitive sodium channels that I talked about earlier and the TTX resistant NAD 1.5 channels. So they're actually all over the heart. What happens there? Very similar story to the other Dravet model with the SCN1A mutation, where we saw an increased sodium current. We saw an increased transient and persistent sodium current. The sodium current, the basis of it was an increase in SCN5A that encodes NAD 1.5, and we saw action potential duration lengthening and an increased RR interval and an increased QT interval. So similar phenotype from different mechanisms, different disease, different genes that are mutated that are modeling the same disease. So Dravet syndrome is a perfect storm. It's a catastrophic illness caused by mutations in SCN1A and SCN1B that both encode subunits of voltage-gated sodium channels. And SCN1B encodes multifunctional beta subunits that are channel modulators and cell adhesion molecules, and the beta 1, the SCN1B null mice are also a model of Dravet syndrome, like the SCN1A heterozygotes. Didn't have time to tell you, but the primary defect in the SCN1B null brains is neuronal pathfinding and fasciculation. It's different than the SCN1As, but it comes out with the same phenotype. But both the SCN1A and SCN1B null hearts have increased sodium current and a prolonged QT interval. Different DS phenotypes through different mechanisms, similar phenotypes through different mechanisms. And in progress, we're looking at now human SCN1B mutant iPSC neurons and cardiac myocytes. So the take-home message that I want to leave you with is that sodium channels are important in both brain and heart disease. Mutations in SCN1A are linked to Dravet syndrome, and cardiac arrhythmias may play a role in SCN1A-linked SUDEP. And mutations in SCN1B are also linked to Dravet syndrome and SUDEP. And we propose different mechanisms result in similar phenotypes through this arrhythmia, so to speak, in the brain and the heart. So this is the result of team science, largely through 3 laboratories. These are people in my laboratory. I've named the ones that really have contributed. Our collaborators, Jack Parent, Miriam Meisler. We also work with Mario Delmar, who was just here, and Pepe Felipe at the University of Michigan. And these are our funding sources. And with that, I'd like to thank you and take questions.
Any questions or comments?
So what is the difference between cadrons in your work? What is how do you explain this?
So there is a new study coming from Al George and Jennifer Carney's work in the same mouse, showing that if you wait a couple of days longer, that the glutamatergic neurons in the same mouse have increased sodium current and hyperexcitability. Okay? So there is a time dependence to this development. There is a neuronal subtype source to this. For instance, they have not been able to show a phenotype in the cortex, and we're looking at neurons that are probably largely cortical. So it could be differences in the area of the brain studied, differences in the time course of development, and Al George and Jennifer Carney have found that there are genetic background differences. So our patients, there are huge genetic background components here, which we may not be able to see anymore. So it's a complicated issue.
And the developmental time course by which this occurs is interesting. And it would correlate rather well with maturation of inhibitory cells in the cortex in the brain.
Right.
So that would point to Cadol's interpretation.
That early on there was a decrease, the disinhibition.
Well, right, so the minute you have
Yes.
a downregulation of sodium current in a particular subset of the neurons,
Right.
hyperexcitability would emerge, and that would correlate with like P20 or something like
Right.
this.
In our iPSCs, though, we never see that the decrease in current. So you may think that maybe we were waiting too long. So we backed up, and we never saw that decrease. Then we said, well, maybe the controls take longer to mature than the DS, and they would get bigger if we went longer. So we went out 10 additional weeks and still saw the increase. So there's a real difference here. But it really could be the area of the brain that we're looking at. And I know that Bill's lab tried a long time to find a phenotype in the cortex, and weren't able to really make heads or tails of it, because as soon as you dissociate the cortex gene, you've got problems. But that could be it. Yes.
For your increased amount of 1.6 and 1.5 that you're seeing, do you think it's either due to just more beta 1 being free and can stabilize the protein that's already there, or do you actually see transcriptional upregulation of those?
In the SCN1B nulls, we see transcriptional upregulation of SCN5A. So, my thought is that we know that the tail of beta 1 can be cleaved in a regulated way and translocate to the nucleus and influence sodium channel alpha subunit transcription. So what if this happens? That under normal conditions, that tail of beta 1 goes to the nucleus in a cardiac myocytes and controls the level of SCN5A that is made. Then when you have no SCN1B, it's a null, then the level of SCN5A can go crazy, and you have more. Now, we need to test that. But it's been shown, at least for beta 2, that that kind of thing happens with SCN1A. So that's not so far-fetched. With the SCN1A cats, those mice, we don't know the answer. We don't know the difference, the explanation for the increase 1.5. But it's very similar in both models.
In these patients, has anybody looked at the sympathetic, parasympathetic balance and things that have to do with neural control of the heart?
So, excuse me, we've started to look at heart rate variability and the autonomic tone. We're analyzing that now. We have not found any basal differences in autonomic tone, but we do see differences just prior to death when we see the bradycardia. What we are making is autonomic neurons from the iPSCs. So Jack's lab has learned to make autonomic neurons, and we'll be recording from those soon, so we'll be able to see that. We're also looking at pharmacological denervation of the heart and pharmacological intervention to look
What
at.
happens if you give beta blockers?
We have the data, we don't know the answer. Yeah, we did the experiment, but we don't know the answer. Yes?
What's the interpretation of the fact that the children seem to be normal for a year and then begin to display these seizures? Is there a normal developmental switch in the expression of some of these alpha subunits?
Yeah. So I think it's like our mice, they're normal until about postnatal day 10, which in a human would be more than that, and then they begin to seize. And part of it, NAD 1.3 is largely an embryonic channel, and that doesn't appear to be affected. Okay, so I think NAD 1.3 is fine, and then later on, when other channels have to come into play, NAD 1.1 comes up later, then you have problems because you don't have the influence of that gene being expressed. So it is developmental. Now, a caveat is all of those studies have been done in rat and mouse. not in human brain or in human heart. And that's what we were talking about this earlier, that those studies need to be really carefully done now so we can understand what those developmental time courses are.
Were the ECG differences in the phenotype in the mice, were they only right before death? Or did you
No.
show some that were?
Yes, so actually we saw ventricular fibrillation early on. We would see them go in and out of arrhythmia, multiple different kinds of arrhythmias. I just showed you the terminal events where we saw that bradycardia. We didn't see the bradycardia happen until the end,
Okay.
but we did see the differences in the ventricular, the VT and VF before that.
And you haven't done anything in the children? The two patients that had, one that had the increases in sodium current, the other one didn't. Have you looked at heart function?
Those children, so we have a collaboration now with Children's Memorial Hospital at Northwestern that is a destination center for patients with Dravet syndrome. And with those pediatric neurologists, they're now starting to measure the ECGs of these patients and the long-term ECGs, so putting Holter monitors on them to actually understand it. You'd be surprised how the neurologists and cardiologists really haven't been talking to each other until recently on this problem, and now they're becoming aware of it and measuring both.
So but you don't have it for those two patients, because it would be nice
Not yet.
given the.
Yeah, we're in contact with the neurologists who got these samples for us, and we're going to try and do that.
And the other question I have is that you showed that there are decreases in the voltage threshold for the action potential in the first part for
Yeah.
the SCN1A. But there's also the depolarization of the resting membrane potential. So if you bring the cells back to control resting membrane potentials, do you still have all those changes in excitability? Or so, in other words, is all the changes in excitability because of the change in resting potential, or do you still have them when you bring the cells back down to?
If we bring the cells back down and clamp and then give them a pulse, then we get the repetitive firing in the DS, but not the control.
Okay.
Okay?
From the same resting potential?
Yes.
Okay.
Yes. Now we know, and we're still pursuing it, that there are changes in potassium currents in these cells. And so this is not just an effect on this particular sodium current.
The voltage threshold from the same resting potential, is it the same?
Sorry?
Voltage threshold? The voltage threshold for the action potential generation. The threshold for action potential generation.
Oh, that's I showed you that's slightly depolarized in the DS patients.
But when you bring it back down, still depolarized?
That's a good question. I don't know the answer. That's a good question. Yes?
Okay.
Oh,
Wonderful question. So you have a very powerful team of collaborators who could also assess arrhythmogenesis and predisposition to arrhythmogenesis at the organ level. So I don't know if you have done or are you planning to do with Pepe Khalifa, let's say, looking at conduction velocity, at the restitution properties of conduction, because
yes.
in the mouse it's really hard to induce ventricular fibrillation.
Oh, yeah.
And what Catherine asked was indeed really fascinating questions, but I understand it's really hard to answer with only 3 mice which died. But knowing what we know now, and Mario just taught us how trafficking is really coupling sodium channels with connexins and with potassium channels. So I would expect to have also changes in connexins, for example. So, and of course, a simple question would be how conduction velocity is affected.
Yeah. So we are doing those experiments with Pepe Khalifa. We are also collaborating with Mario Delmar to look at the connexin, the PKP2, the cankrin. What's the structure of that macromolecular complex in these mice? Is it different? Mario's also doing the attachment right at the T-tubules, right at the intercalated disc to ask those important questions. And finally, because the mouse is so difficult to look at for cardiac properties, we're generating a rabbit model to go with this too. And hopefully that will be a SUDEP model. And we can do these experiments again.
Is there any evidence for structural heart disease in this children?
There's none.
All right, I think we have to close this. Thank you so much.
Thank you.